This presentation will be over a genetic approach to antibiotic discovery from bacteria. Our lab studies two types of bacteria, Streptomyces and Bacillus subtilis. These model soil bacteria undergo many complex reactions caused by environmental factors and competition from neighboring bacteria. The stress caused by competition and environmental changes can result in the production of specialized secondary metabolites as shown. Secondary metabolites produced by Streptomyces have previously been characterized as antibiotics that currently are used in many fields of medicine. These listed antibiotics are only a few of the antibiotics produced by the genus Streptomyces. They have many uses in the medical field, including treating tuberculosis, methicillin-resistant Staphylococcus aureus, and meningitis. This genomic diagram of Streptomyces MG1 illustrates the large number of unknown biosynthetic gene clusters in a single species of Streptomyces, each of which could potentially produce useful antibiotics. The horizontal line represents the entire genome, while each vertical line represents the locations where genes encode antibiotic synthesis and other secondary metabolites. As you can see, there are only two identified secondary metabolites in Streptomyces MG1. Taking into consideration the number of Streptomyces species that have not been fully researched, this genus holds a treasure trove of possible antibacterial compounds. However, discovery of new antibiotics from this genus is historically difficult due to the low production of specialized metabolites in laboratory conditions and the lack of isolation and detection tools. To combat this problem, we propose using CRISPR systems to target specific gene clusters in order to reduce or completely stop the production of the corresponding metabolite. Based on transcription analysis, the cluster 9 biosynthesis gene was chosen as a candidate for characterization. This gene cluster is suspected to produce a thiopeptide. CRISPR-based systems of gene editing enables us to target specific individual biosynthetic gene clusters. The standard CRISPR-Cas9 method allows us to manipulate and delete the biosynthetic gene cluster, while the CRISPR interference, or CRISPR-I-based method, allows us to silence expression of the targeted gene. Both methods are used in tandem to optimize the data collection. Following the generation of both mutant strains, a complementation assay was performed on the deletion strain to confirm that there is no other ulterior effects of the Cas9 during the conjugation. A series of data collection and analysis techniques were performed on the mutant and wild-type strains. Since we are searching for unknown metabolites, we will use two basic methods to look for evidence of silenced phenotypes in our targeted biosynthetic gene cluster. The first will be sporulation-based techniques, meaning we will run a series of plate assays to determine any noticeable differences in development or phenotype among our engineered strains. The second will be direct metabolite identification, in which we will extract the organic compounds from our sporulated strains and run these organic compounds through several compound detection assays in order to find the metabolite's exact weight and structure. We performed a development assay on the engineered strains as shown. The pictured samples were grown at 30 degrees Celsius for 96 hours, giving them ample time to sporulate since Streptomyces MG1 sporulation is usually completed at 72 hours. The wild-type strain is the control, while the deletion strain acts as a control for the mutant phenotype. Following that, the CRISPR-I strain tests for any developmental effects of the CRISPR-I method, while the complement strain genetically restores the deletion mutant to wild type, letting us see if there is any effects that are specific to the mutation process. There was no determinable phenotypic differences among the strains. This suggests that the targeted gene does not affect developmental aspects of this organism. We also performed a competition assay on the engineered strains. We used a linearmycin-resistant strain of Bacillus subtilis as the competitive organism. This competitive organism was chosen because its sliding motility is known to be affected by many of Streptomyces' secondary metabolites. However, there was no noticeable difference in phenotypes among the strains. This is our direct metabolite identification technique. Each of the deletion and wild-type strains were grown to sporulation on pH-balanced media and extracted with organic solvent. The organic extractions were ran through MALDI to determine any metabolic differences. In theory, the wild-type strain should have a peak that is missing in the deletion strains that correlates to the targeted gene product's mass. There were two possible peaks identified as candidates for the target molecule, a peak at 1355 and a peak at 1597. Both peaks are within the mass range of thiopeptides, which is what our target molecule is suspected to be, and both appear to be absent in the deletion strain. Notice that this is a comparison of two deletion strains and the wild-type strain. The CRISPR-I strain was omitted because it has yet to be analyzed using MALDI. In conclusion, we have shown that a genetic approach to metabolite identification in Streptomyces is effective in targeting a gene of interest. We have evaluated the phenotypic differences caused by our target gene cluster, and we have found mass candidates for the metabolic product of our target gene. Going forward, we will use LC-MS to confirm the identity of the metabolic product and gain insight into its structure. I would like to thank Chengxi Zhang, Dr. Paul Strait, Keliani Drosela, Larry Dengo, Sandy LeBont, and the Department of Biochemistry and Biophysics at Texas A&M University.